Hello friends, welcome back to my YouTube channel Nasreen Padasale. So this video is all about the question that has been asked under unit number 6 plant physiology both for shift 1 and shift 2. So the first question is that digerent protein predominantly play an important role in biosynthesis of. Actually, this protein play an important role in secondary metabolism of plant, and this particular digerent protein are mainly involved in biosynthesis of lignans and flavolignans. Both the product that is plant secondary metabolites has been produced under the responsibility, under the control of this digerent protein. Apart from this digerent protein have and play a major important role. Okay, so please don't compare your lignans with lignins. Both are of course a plant secondary metabolites, but lignan is actually small and they are soluble in H2O, whereas lignin, that is LIGNIN, it is insoluble in water because it is highly polymerized structure. Okay, so digerent protein play an important role in biosynthesis of your lignans only. So the next question. Photochemically generated ATP is consumed in which one of the following phases of Calvin-Benson cycle? It is your C3 cycle. So if you remember this particular diagram, you can be able to easily answer the question. So in this particular diagram, this Calvin cycle consists of 3 stages: carboxylation, reduction and regeneration. So carboxylation reaction doesn't require any ATP to convert this particular ribulose 1,5-phosphate to 3-phosphoglycerate, friends. Okay, whereas the reduction phase, where your 3-phosphoglycerate will be reduced to glyceryl 3-phosphate, and your regeneration phase, where the glycerol 3-phosphate will be regenerated into ribulose 5-phosphate. Okay, so this particular regeneration stage and the reduction stage alone require the ATP. So if you remember this diagram, then you can easily answer the question. So the answer is that photochemically generated ATP is consumed during reduction stage and regeneration stage of the Calvin-Benson cycle. Okay, so next, in which of the following stages of Arabidopsis embryo do the visible distinction between adaxal side and abaxal tissue of cotyledon become initially apparent? So you should know plant embryo will be undergoing important stages like the first 2-cell stage, next 8-cell stage, next globular stage, next triangular stage, next heart stage, next torpedo stage. Okay, this particular adaxal means upper surface of leaf or cotyledon, whereas abaxal side means lower surface. Okay, so both the cells present in upper side and lower side, they have a different morphology. Like if you take the cells present in upper side, it will be elongated in nature, as well as it will be tightly packed with many chloroplast content. Whereas if you take the lower side, the cells will be rounder in shape and as well as it is loosely packed. So this question has been asked under leaf patterning. So keep on updating about leaf patterning in your upcoming CSIR preparation, friends. This is similar to your dorsoventral polarity in your animal. So the question is that what stage of the plant embryo does the upper side and lower side of the cotyledon become initially apparent? So it is initially apparent in torpedo stage, friends. First, the cotyledon primordial will be generated in a heart stage. So then heart stage will be followed by the torpedo stage. The primordium will elongate and form this particular abaxal side and adaxal side. So it will be visual in the particular torpedo stage. So this particular thing and all, only if you are revised the recent updated article, you can be able to answer this question, because even in Ty's book I could be able to find this particular answer. Only in particular thing in this particular paper only I just found this particular answer, friends. So keep on updating yourself with the recent research article that has been published under that. Next, following are the statement regarding the root growth and differentiation in plant. So first option, so they had given 4 options, that we need to select which are the option correct. Root hair, endodermis, xylem, phloem reach maturation in the elongation zone of developing root. So the developing root consists of 3 zones: maturation zone, elongation zone and meristematic zone. So they had given root hair, endodermis, xylem, phloem will reach the maturation in elongation zone of root. So you can see the elongation zone of root. So root hair will be maturated in the maturation zone. Root hair is present in the maturation zone, whereas your endodermis, xylem, phloem will be matured in the elongation zone. So root hair will be matured in the maturation zone, but here they had given it will reach maturation in the elongation zone. No, your endodermis, xylem, phloem will reach the maturation at the elongation zone in the developing root. Next, root epidermis cells that are incapable of forming root hair are called as atrichoblast. So here you can be able to see the outer part is called as epidermis, and not all the epidermis is present with the root hair. So the root epidermis is regularly spaced between trichoblast and atrichoblast. So here you can be able to see a root hair, whereas a cell next to it is not having a root hair. So those cells which will giving rise to a root hair is called as trichoblast cell, that is tricoblast, whereas those cells that doesn't give a root hair is called as non-hair cells, and that is called as atrichoblast. Next, the root quiescent center is present just above the root hair. So if you remember this particular structure, so this structure I just taken from Price book, friends. So root hair, above that you can able to see quiescent center. This quiescent center is nothing but it's present in a root only, and they have a low mitotic activity, as well as they will be rarely dividing. If you ask me what is the function of this particular center, means this particular center is a hub of stem cells. So whenever the plant is facing any kind of injury, definitely from this particular center only the stem cells will be provided, and plant will be regenerating its root apical meristem. Okay, so option C is also correct. Next is Arabidopsis auxin transporter, that is ABCB4, which is a transporter protein. The full form is that ATP binding acid play an important role in root hair emergence by maintaining the intracellular auxin concentration. It is absolutely a correct thing, friends, because this particular ABC4 is involved in actin transporter, play a role in root hair. So where is the presence of root hair? So root hair is present over here. So here the root hair is present, and here I had given you a total list of ATP binding protein. Like if you take the ATP binding protein 19, that is ABCV19, it is present important role in hypocortical, like it is playing an important role in producing this pericycle bundle sheet. Whereas if you take this ABCB15, it is present in the vascular bundle like that. Whereas if you take ABCB19, again this ABCB19 is present both in cotyledon and hypocortical. Same like that, ABCB4 is present in your root epidermis of root, as well as ABCB4 is also present in the root apex. So this is also a correct option. So these are the important thing. Okay, so the next question is with regard to the important thing. So first, phenolics is having a aromatic ring with the OH group. Next, alkaloid will be having a N containing group. These are easy things, friendly friends. Terpenoid is having a 5-carbon isoprene unit. That's a basic structural unit of terpenoids. Next, cyanogenic glycoside. This cyanogenic glycoside consists of a glucose unit which will be attaching it to a O beta D glucosyl linkage. Next, following a certain statement with regard to phytochrome interacting factor. Phytochrome interacting factors are a family of protein that are regulating the photomorphogenetic response in the plant. So they given 4 important statements, and they are asking which of the following combination of statement is correct. Okay, so the first thing is that these PIFs, that is phytochrome interacting factor, promote scotomorphogenesis. So scotomorphogenesis means development of plant in absence of light or as in the dark. Okay, by serving as a transcription activator of dark induced things. So this phytochrome interacting factors are transcription factor that will be activating a dark inducing. This is absolutely a correct option. So option A is correct. Next, these PIFs are interacting with the PR. It will get phosphorylated followed by degradation via proteasome complex. So this PIFs, that is phytochrome interacting factor, once they complete their duty, they will be undergoing degradation, that also by this ubiquitin proteasome complex. But this PIFs doesn't interact with the PR. It will be interacting with the PFR protein, that is PR. Once they get activated, they will be converting into a PFR, which is an active form of phytochrome. So option number B is wrong. It is interacting with PFR. Next, degradation of phytochrome interacting factor takes place in the presence of light. Of course, once the light has been present, then ultimately this PIFs are not being required, because other protein that are involved in photomorphogenesis will be active. So in the presence of light, this PIFs protein will start to degrade. Next, PIFs induced genes are not expressed in light. Absolutely correct. They are expressed only in dark condition. Next, following are the statement with regard to tracheal element of vascular plants, friends. So tracheal and vessels are the element that are helping in the water transport. Okay, so both the tracheates and vessels together, they are called as tracheal element. If you take trachea, it is present in a xylem of angiosperm, gynosperm and fans, whereas vessels are only present in angiosperms. Okay, so first thing is that xylem tracheates are highly elongated, tapered cell that conduct water. Absolutely correct. If you see the structure of tracheate, it is taper, which means narrow at the bottom of narrow towards the bottom. Okay, just like your ice cream cone. Next, if you take the structure of vessel, it will be shorter as well as wider at the bottom. Okay, so option number A is correct. Next, xylem vessel element are less elongated and narrower than tracheates. No, xylem element are highly elongated and they are wider, and they are not narrower, they are just shorter when compared to tracheate. Next, angiosperm curve, both tracheates as well as vessel element. Angiosperms have only tracheates, angiosperms may have both tracheids and elements. So it's absolutely a correct statement. Angiosperm is present both in tracheids and vessel element are present in angiosperm. So angiosperm have both option, like it can either use tracheids as well as vascular element for conducting water. So option C is also correct. Next, vessel element are the only tracheal element present in almost all gymnosperms. So vessel element are present only in angiosperm. It's not present in gymnosperm. Okay. So option number A and option number C are correct. Next, during light reaction in photosynthesis, electron is transported in the electron transport chain and produces ATP and NADPH product. Following are the statement regarding ETC during light reaction. So here I had given you a diagram for your reference. So electron from P680 moves first to quinone. It is absolutely a wrong statement. So first the electron will be moving to pheophytin, then only it will be transferring to quinone. So option number A is wrong. Next, P750 can receive the electron from plastocyanin. Absolutely correct. P750 will be getting the electron from plastocyanin. So PC. Next, NADPH is produced. NADPH is produced at the end of light reaction. Absolutely, the outcome of light reaction is the production of large number of NADPH. Next, the hydrogen ion produced during the light reaction get concentrated in the thylakoid domain. So here, the hydrogen ion will be highly concentrated in the thylakoid domain. Only if there is any necessity, then this hydrogen ion in the thylakoid domain will be coming out from the thylakoid domain and it will be leading to the production of ATP. Okay, so hydrogen ion, that is proton, will be highly concentrated in the lumina of the thylakoid. Okay, so next, the option number B, C, D are correct. Next, a researcher obtained an Arabidopsis mutant defective in stridolactone. So particular plant is not having a stringolactone synthesis, the novel plant hormone. Okay, following a statement were made regarding the mutant phenotype. So whenever you are coming across the name novel, you just refer the recent article. Okay, so you should know the function. Just list down the function. So just write the function of stringolactone in your paper itself. Only then you can answer correctly this question. First, shoot branching get enhanced in the mutant plant. So first important function of this stringolactone is that the plant which is having an active form of stringolactone will suppresses the shoot branching, that is secondary branching in shoot. Okay, so this option is absolutely correct because this particular plant is mutant and it does not have stringolactone. So the shoot branching does not get suppressed, it will be getting enhanced. So option number A is correct. Next, hyphal branching of AM fungi get enhanced during colonization in mutant plant. Okay, so if you take the function of the stringolactone, this stringolactone, that is SL, will induce the hyphal branching. But here what they are saying, the hyphal branching get enhanced. But here this plant is not having stringolactone. It is mutant. Mutant means what? It doesn't have stringolactone itself. It doesn't have this SL hormone at all. So at this time point, it cannot able to enhance. It should suppress only because SL is not present. So option number B is absolutely wrong. Next, shoot branching get inhibited in mutant plant. Absolutely wrong statement because shoot branching will be enhanced in mutant plant. Only if the plant is having an active form of XL, it get inhibited by the normal plant. Next, germination of seeds of parasitic plant is prevented near the mutant plant. Absolutely a correct answer because a plant which is having an active form of stringolactone will lead to an active germination. That stringolactone will stimulate the germination of seeds. But here they are giving germination of seeds of parasitic plant will prevent near the mutant plant. Absolutely correct because mutant plant will not have the stringolactone hormone, so that the germination of seed is prevented. Okay, next is with respect to shift 2 question. Which one of the transcription factor is important for delimiting the meristematic and elongation zone of fruit? So you should know the function. If you know the function itself, you can able to easily answer this question. So the answer is that this plethora gene or plethora transcription factor is important for distinguishing between the meristematic zone and elongation zone of root. Okay, so here you can able to see there are 3 zones of root: meristematic, elongation and maturation. Okay, so plethora gene is important for delimiting 2 zones of root, whereas if you take sarco and short root, both the genes or transcription factor, they are important in dividing your endodermis and cortex. So they are important in distinguishing between endodermis and cortex, whereas if you take this plethora gene, it is important for distinguishing between meristematic and elongation zones of root. Next, the speechless gene, this particular transcription factor is involvement in somatal development. Next, Following on the certain statements related to seed maturation. So seed maturation involves the accumulation of storage products like starch, lipid and protein. Absolutely a correct statement. So generally if a seed is maturing, then it generally involves the accumulation of storage products like starch, lipid and protein. So option number A is correct. Next question is with respect to the large number of chaperone including the family of late embryonic aberrant protein play an important role in desiccation of seed. So this LEA protein, this protein, they generally accumulate in a seed in a large amount of volume when the seed is undergoing a seed desiccation process. So just seed desiccation generally occurs in the later part of the thing. Apart from the seed desiccation process, this late embryonic abandoned protein or involvement in plant tolerance to abiotic state. So option number 2 is called, so that is option number B is also correct. Next, moisture content get reduced with the maturation. So as a seed is getting matured, it is entering into the later stage. In the later stage, the desiccation of seed will be occurring. Next, ABA and gibberelling both promote seed germination. Of course, ABA promotes seed germination, but if you keep gibberelling, they will promote seed germination, not seed dormancy. Okay. Next, these with respect to biosynthetic pathway of terpenoid, friends. So they had given a list of things and they are asking which enzyme is involved. So while studying for upcoming CSIR examination, please note about the enzyme for the biosynthetic pathway. Generally, we will be skipping up the enzyme and all, friends. So please make a note of it. Biosynthesis of two basic 5-carbonate unit, that is synthesis of isoprene unit, it is done by this HMG-CoA synthetase enzyme. Next, repetitive addition of C5 unit. So you should know the terpene are classified into hemiterpene if it is consist of one single basic isoprene unit. So terpene is generally a polymer of isoprene unit only, friends. So if another C5 unit is added, then it will become C10, then it is called as monoterpene. Sesquiterpene. When there is another addition of C5 molecule to this monoterpene, it will be converted into sesquiterpene. So this repetitive addition is being managed by the enzyme called perennial transferase. Next, formation of basic terpenoid skeleton, it is done by an enzyme called terpene synthetase, and after that modification of terpene synthetase. So if there is any need of modification of your terpene synthetase, it is done by this P450 monooxygenase enzyme. Next, following on the statement with regard to nitrate transporter in plant cells. So there is a question that has been asking about nitrate transporter in plant cells. So they had given 4 options, and in that they are asking which of the following statement is the correct option. Okay. So first, AT means Arabidopsis thaliana. Okay. NRT means nitrate transporter. First question is that nitrate uptake display 2 saturable phases. So first phase is the high affinity system and next phase is the low affinity system. High affinity system generally expressed in a micromolar range, whereas if you take low affinity system, it will be expressed in a millimolar range. Of course, the option number A is correct. Next, Arabidopsis, that is AT-NRT 1.1 is a dual affinity nitrate transporter. That is, it can act either as a low affinity nitrate transporter or it can act as a high affinity nitrate transporter. It is also absolutely a correct option. Whereas if you take this AT-NRT 1.2, it is participating in high affinity uptake. No, AT 1.2 participate in low affinity uptake, whereas if you take AT, NRT 2.1 and 2.2, they are involved in high affinity option. So both the C option and D option are been ulta, friends. So only A and B are correct option, whereas C and D are wrong option. So AT, NRT 1.2 is involved in low affinity uptake, AT 2.1 and AT 2.2 are involved in high affinity uptake. Next, following our statement regarding to photorespiration pathway, the first 2-carbon compound synthesized by the action of riboscope in the chloroplast is glycolate. So, the first 2-carbon compound synthesis is your phosphoglycol or phosphoglycolic acid. After that only this that is P glycolate will be synthesized to glycolate. So, they are giving first 2-carbon is there, not a glycolate, it is phosphoglycolate. Okay. Next, option number A is incorrect. So here they are asking combination is incorrect only, friends. Next, glycolate exit the chloroplast and enter into peroxisome. It is absolutely correct thing. So glycolate which is produced in chloroplast will be exiting and making way to peroxisome. It is correct option. So here they are asking only incorrect option. So this is correct option. So just leave B option. Next, glycolate is that is synthesized during C2 cycle enters the chloroplast from mitochondria. Okay, so you can able to see glycolate which is synthesized in chloroplast doesn't make it way to mitochondria. It is getting made to way to peroxisome. In peroxisome, the glycolate will be converted into glycosylate, and glycosylate will be converted into glycine, and glycine will be entering to glycine will be entering to mitochondria. So option number C is also incorrect. So the correct option, that is which one of the following combination incorrect, is option number A and option number B. Option number D is correct because glycine will be transported from peroxisome to mitochondria. If you remember this diagram itself, then you can easily answer this particular question, friends. The following are the statement with respect to photosystem and structure and function involving light dependent reaction of photosynthesis. Okay, so the first antenna or light harvesting, they are saying which of the following statement is incorrect. So please make sure what they are asking, whether they are asking which of the following combination of both statement is correct or incorrect. Then only you should read the option. Okay, so read the question twice or thrice and read the question completely. Okay, so this is how many students are leaving their opportunity to clear CSIR examination. So antenna or light harvesting complex absorbs light energy and it will be transferring to reaction center. So that is an correct option, friends. So option number A is correct, but here they are asking incorrect option. Next, first electron release from P680 is transferred to QA. Absolutely it is not. It will be transferring, it will not be transferring to QA, it will be transferring to pheophytin. Okay, so we can select B option, that is incorrect. Next, D1 is a protein subunit of plant photosystem 2 co-complex. It is encoded by gene PSPD. That is absolutely a wrong option because D1 is a protein which is actually involving photosystem 2, but the gene which encoded by encode this D1 protein is not PSBD, it is encoded by PSBA protein. So this PSBA protein means photosystem 2 reaction center protein B. It is encoded by plastic gene, that is this gene is present in chloroplast genome because this photosynthesis all are taking place in chloroplast. Next, they are asking questions with respect to glycophages and halophages. Glycophages are salt sensitive plant, which means they cannot able to tolerate even a little amount of salt is getting increased. Okay, as halophages are salt tolerant plant. The following statement were made to explain the difference between glycophages and halophages. Okay, so glycophages, they enhance the uptake of ion. So here they are asking what are the combination of all statements are correct like that, friends. So glycophages enhance the uptake of ion. No, it is absolutely a wrong option because already this glycophages is a salt sensitive plant. So then how they can able to enhance the uptake of ion? They won't enhance. Instead, they will be actively pumping the ion back to the soil. So option number B is correct. Glycophages can be pumped the ion back to the soil. Next, halophage. Halophage have the ability to resist the net ion uptake in shoot. So from root only salt and many substance will be transferring to shoot. What does halophage mean? Halophage means salt tolerant plant. This plant has an ability to prevent the uptake of ion to shoot from root. Okay, so option number C is correct. Halophages have a greater capacity for vascular sequestration of ion. So ion transport is actually an important mechanism for determining the salinity tolerance in plants. So there are two mechanisms by which ion will be transported. One is across the plasma membrane, next is with respect to vascular membrane. So this halophage plant, what they will be they will be having a greater capacity. So once they find a particular ion getting increased means they will be sequestered, they will be sending all the ion to vascular portion where the ion will be stored. So this is what with respect to both the things. So thank you friends, thanks for watching this video.